Okay, so we'll continue with the second part about the sequencing itself. And so I'm sorry, I will do a story again, but it's always interesting because you are catching some flavor of how these things have been developed. And the big name in the history of the sequencing is definitely Fred Sanger. And in fact, this guy deserved probably 3 Nobel Prizes, and he got only 2, which is a pity. In the 50s, he started by resolving the issue of doing the protein sequencing, and he got the Nobel Prize for his capacity to decode the insulin sequence, which was a breakthrough at that time. And so what is interesting in that case is that the DNA was not possibly sequenced at that time. It was such a complex thing to work with that nobody was considering that it would be possible. If I have to make a comparison, this is probably similar to the difficulty that we have now today to identify the glycosyl moieties on the glycoproteins. The level of complexity at that time was probably the same. And what was particularly difficult was the fact that the DNA appeared as a very large and very long molecule. And so even if the three-dimensional structure of it was resolved in 1953, the capacity, the experimental capacity to read was not available in the 1950s. And so this is through the RNA that the nucleic acid became sequenced. And it was not on DNA, but at that time, it was kilograms of transfer RNA that were generated. And it was really with a huge amount of material that it was possible to achieve the first sequencing of a nucleic acid that corresponded to the tRNA for aniline in Saccharomyces cerevisiae. And at that time, what was done was a combination of ribonuclease treatments and assessment of nucleotide composition. And so by doing a series of digestion, people were able to consider what was the ratio of the different nucleotides and get the full sequence. So it was really not something very easily done. This was particularly beautiful to achieve that, but Ole et al. Ole got the Nobel Prize for it. And what I said is that at the same time, Sanger started to develop an alternative to the technique in which he was using some radio labeling. So he was able to follow the partial digestion using the radio labeling of the RNA. And he was resolving that by 2D fractionation, so the old type of genes where you are resolving the charge and the size of the fragments. And so at nearly the same part of time, he generated information not only about transfer RNA, but also about ribosomal RNA. And this is his method and not the method, the very harsh method by Ole that was used to resolve these other sequencing. In the early 70s, the first results were generated on DNA, and this was done by Fierce Laboratory that did the sequencing of the code protein of the bacteriophage MS2. And then they were able to get the full genome of the bacteriophage, and it was always by 2D fractionation. So these were very tedious methods, and again, the capacity to develop specific oligonucleotides that were priming the DNA polymerase, the incorporation of radioactive nucleotides to infer the different orders of nucleotides. It was done, but only on very short stretch of DNA, and that was very demanding in terms of the experimental procedure. In around 75, some of you are perhaps remembering this type of big gel that we were running in the lab at that time. So there were very large resolution electrophoresis in which we were resolving polynucleotides according to their length in polyacrylamide gel. So it was this type of picture that you are getting. What turned out, so there are different protocols. The first one that appeared was by Maxam and Gilbert, but then the method developed by Colson and Sanger became dominant. In the two cases, what they did is that they were synthesizing with DNA polymerase starting from a primer, and they incorporated radiolabeled nucleotide. And during the second round of polymerization, they were adding one part of the mixture, so not the ACGT, for instance, but only the A, or only the C, or only the G, or only the T. And with that, they were able to compare the stop of the synthesis that were generating different bands at different stop position that did to the position where the nucleotide was missing. And so you can do that for the A, for the C, for the G, for the T, and each time you will have a different position, and at the end you are coming to the A, T, G, C, A, J, C, G, T, etc. And you recover exactly this sequence. In the case of the Maxam and Gilbert, the situation was slightly different. What they introduced was a cleavage by specific molecules. So, for instance, they were using a treatment with chemical like dimethyl sulfate, formic acid, hydrazine that was cutting at the C plus T, at the C, at the A plus G, etc., or in presence of sodium chloride. And with that, they were doing a sort of composite picture, and they were also able to generate the sequence. So that's the old age, and I would say that a large part of the effort started like that in the 70s. A major improvement was in 77 when Sanger discovered and developed the chain termination, also called the dideoxy technique. And so what happens is that the dideoxynucleotide lack an hydroxyl group that is required for the extension of the DNA chain. And therefore they cannot form the bond to extend to the next DNTP. And so by using a mix of DNTP, the dideoxy NTP during a DNA extension reaction, there will be a start of the reaction at the place where the dideoxy has incorporated. And again, you will have a ladder with a different chain that you can identify. So initially it was done with radiolabeled dideoxy DNTP, but then it was replaced by fluorescence, and you have the characteristic pattern that you have probably seen in the textbook that were used for the large effort of sequencing. And so after that, the story, we are still in the beginnings, but there were improvements after the incorporation of the fluorometric base detection, after the introduction of capillary electrophoresis instead of big gel electrophoresis, automation come out, and it was possible to generate with that sequencing machines that were able to read fragments of up to 1 kilobase. It was quite interesting. So this led to the first shotgun sequencing. So by taking DNA libraries or cDNA libraries, it was possible to generate information about quite a significant amount of genomes. It was sometimes difficult when you were finding fragments that were difficult to clone or that were isolated in highly repeated sequence. But that led through the assembly of the different fragments to an in vitro assembly to get long contig. So this epoch was marked by the introduction of the polymerase chain reaction. So I have not to introduce that because even the deepest informatician has seen Jurassic Park or sort of movie like that. So we are all aware of the polymerase chain reaction. And if not, we have lived during the pandemic time, and so we know what is the PCR. And what was also quite important was all the action around the technology of recombinant DNA to generate nice high concentration of pure DNA species, and also all the enzymes in a way that were well described yesterday in terms to optimize the quality of the enzyme. Among them, for instance, the cleno fragment of the DNA polymerase played a big role because it was very efficient to work with dideoxy DNTP. And so its creation after managed protease digestion was very useful for the progress of the field. And so the identification of better polymerase or the creation of new type of dideoxy sequencer was characteristic of that time. And then a new concept came out, and it was a work by Paul Nieren and colleague that developed after that first time a concurrent large-scale sequencing, and this one was a different concept by comparison with the dideoxy sequencing. And what was interesting is that the pyrosequencing is a sequence by synthesis technique in which the direct activity of the DNA polymerase is used to generate an observable output. And so the general idea is that when you incorporate a nucleotide into a sequence, you will generate a pyrophosphate, and this pyrophosphate, if it is put into the presence of an enzyme called sulfurylase, it is able to convert back in the pyrophosphate into ATP. And a second enzyme, which is called the luciferase, is able to emit light in the presence of ATP. And so the emission of light will point out the presence of a specific nucleotide. And if you have added an ATP or a GTP here to generate a pyrophosphate to generate ATP, then you will have a peak of light, and this is exactly what you will detect. And so then you can wash out with an apirase, you clean the substrate, and you can start the next turn of synthesis, and recurrently you will generate a full sequence. And this pyrosequencing was licensed to a company called 454, and this became one of the first major successful next generation sequencing product, which was then bought by Roche, that commercialized it for several years. So the system was interesting. It was not so good when he was meeting some repetitive sequence, so multiple stretch of ASCGT. That was not very efficient, but otherwise it has been very useful for several years. There were some improvements that were introduced, and again, it's always of interest to see the evolution of the technology. When we were considering the Taq polymerase, it was to generate very small volume of reaction. With the next generation sequencing, we were directly generating tens of milliliters of PCR reactions, and that was a different different type of experiment. At that time, it was also important to keep the clonality of the sequences. And so when you are doing PCR on fragments that are very heterogeneous, one solution to keep this heterogeneity and to allow the amplification, even if you have different composition or different type of sequences, is to individualize the molecules that are interesting. This is a beautiful concept that makes that leads to the creation of multi compartments in which you can do that. And the general idea to get it is to prepare a sort of mayonnaise in which you will have an oil that will cover tons of multiple droplets of aqueous solution, in which you can have the DNA that interests you, the microbeads on which the DNA is attached, the enzymes that are necessary for allowing the amplification, and so the DNTP to allow also this amplification. And if you are doing this sort of reaction in water in oil emulsion PCR, as we say, in that case you are able to generate multiple clones of your molecules of interest that are, for instance, bound to a bead, linked to a bead, and in that case you have the same molecules that are covering the bead, and you have not a mixture of them. So when you have done this sort of amplification, and when people have done this sort of amplification, they were able to load them onto picot titer wells that were associated with the instrument. And all the sequencing that takes place was taken place with the same molecule that was covering the bead. And so you can have a very efficient sequencing with that. This type of pyrosequencing allowed through the coupling with a CCT camera allowed the production of 400 to 500 base pairs reads and to cover millions of wells that contain these clonally coated beads. And so this was the one technology that was largely used in the beginning of the 2000 to the 2010, and in 2007, for instance, the DNA of late James Watson was sequenced with this technique. And it was the first trend that became so obvious that sequencing even of a complex genome like the human genome will turn into something very simple and that can be achieved in a short period of time. You can compare the size of the laboratories in which the genome, the first human genome version has been sequenced, and the current laboratories that are doing human genome sequencing. And in one case, you have a huge place, a huge plant with hundreds of instruments, while now you have only one machine that is taking place, a very short place like this desk. So it's always interesting to look at what was going on, and because there were other technologies that were concurrent. There is a very similar technology to the one developed for Roche and 454 that correspond to the ion torrent. In that case, it was a pH modification that was generating and not light. It was already using CMOS technology, so that was interesting to have yesterday the presentation of the synthesis, because for the sequencing, the same type of questions have been raised. The sequencing was very fast, but there was some problem linked to the homopolymer, which was the same for the 454 and less obvious for Illumina. But that technology was supposed to replace the high throughput instruments, and it was expected that Life Technologies, that has been the major company to develop the Sanger sequencing, would be able to develop a large capacity sequencer. And unfortunately, their miniaturization failed because, as I explained yesterday, there was a problem with the pH that diffused very rapidly, and so it was difficult to control the pH gradients around the different spots, and they were never able to reach a similar throughput as the other company. A second effort was developed by Apply Bioscience with a technology that was quite different. It is called SOLID. I just mentioned it because it was the first next generation sequencer that we had in the lab. And it was just to share with you my nightmare during years. It was based on ligation. And so this was a very complex picture in which you were not detecting one nucleotide by a dinucleotide. And so the quality of the reading was so poor that when we were comparing our data with people working with Illumina, they were laughing at us. And so we moved to Illumina then, and the story became different. I'd like to mention also the company called Complete Genomics, because they introduce a technology called DNA nanoball. That the story of the Complex Genomics company was also very bad, and they finished their activity rapidly. But you will see that there are some new companies that are reviving the concept of DNA nanoball. The idea, excuse me, the idea is that if you circularize the DNA that you want to sequence, and that you use a DNA polymerase, you can generate several copies by round circle amplification. And so you can have an amplification of the signal, which is a very common technique that is now used. And you will see the results with a new type of sequencer called AVITI, and the results are now very interesting. But at that time, it is Illumina that became the major player in the market. Until now, I think that they represent 80 to 90% of the sale in the field of the sequencing. So their work was marked by the replacement of emulsion PCR by a bridge amplification. And so the idea was to have two types of oligo on the surface of a glass slide, and on which it was possible to hybridize a fragment that you want to sequence in red, and the white can hybridize with the white, the black with the black, and you see that you will create by several rounds of amplification several copies that can be generated of this fragment that will be localized in a very close proximity. And this was linked to a technology of sequencing by synthesis, where a fluorescent DNTP was incorporated to the system, and there was a continuous reading at different. So if you want to have a very simple explanation and about the difference that exists between the regular Sanger sequencing and what we are doing there, is just that we are inventing a sequencing that is not in one direction but on the surface. And so instead of having a measurement just in one direction with the size of the different fragments and the different base that we were reading before, like here, where we have for one fragment the sequence that is given. In that case, what we have is that we have a picture in which we have different spots on the surface, and at time 1 you have a first base, at time 2 you have a second base, at time 3 you have a third base, and so you will generate the full picture with these different steps. And so the initial work by Illumina came with qualities. Well, they were generating quite small fragments, less than 50 nucleotides, but it was in two directions, so they were able to generate some interesting information, and the throughput was interesting. And so rapidly the throughput was improved, and they are now able to generate, well, I'm not even sure that it is not even more, but with the largest NovaSeq, you are probably over 16 to 20 terabytes per run, which can be generated in less than 2 days. So this, I will perhaps not spend a lot of time. I think that, I don't know if we will share the slides, but we can probably do that. So I can perhaps save some time to make all the comparison between the different technology. And I will review instead this technology, and you will be able to see that afterward. So the Illumina is a company that has been developing an expertise for short read, and they are not very good at long read sequencing. So they are trying like crazy to develop the approach to generate also long reads. And this is the contrary with the companies that are generating long reads that want also to make short read, because they want to have the full market. So the general idea is that they are developing at the moment, but this is not yet available completely, I guess. But they introduced during an enzymatic step that is called tagmentation, some mark into long fragment of DNA, and this is used then for when they are doing some short read sequencing afterwards to get some indication about the position, the relative position of the different fragments, and that helps them to generate a more precise assembly of the fragments. We can discuss that later on, but today I think that the advantage of combining long read with short read to get full sequence information is becoming so clear. I don't know if, what will be the extent of results that will be generated by that, but people have already worked a lot on alternative. And I don't know if there is room still for this sort of thing. So the kind of sequencer is provided by Illumina is shown here. This is a small one, what is called the NextSec 2000, which is already generating a quite large amount of output. And the larger one is shown here. So I wanted to spend a few minutes on the AVITI sequencer and to go directly to the point. I can just show you that compare, and again I will not spend too much time to comment this graph, but this is just to say that today what is crucial is the cost per base, per gigabase or whatever that you can get with your sequencer. And so this is something that is followed very carefully by Albert Villela for years, and you can find that on the internet. This is not necessarily the last output, but he has generated, but it gives us a flavor. And what is very important is that when you look at the cost, some technology are definitely much cheaper than others. What happens is that the technology developed by the BGI are clearly cheaper, but not very far from that. There is this machine called AVITI, and it compares particularly well with the type of sequencer like the NextSec 2000 that is found in a lot of different labs. So if you compare, you can reach a similar level between the AVT and the largest the largest sequencer from Illumina, and so that makes that particularly attractive, and that makes it also particularly attractive because in terms of the quality of the sequencing, we will see that there is a level of quality that is probably better than with Illumina. And so this was published in Nature Biotechnology. The idea is to combine what I already discussed, the Polony, so the rolling cycle amplification, with a new thing that is called the avidit, that corresponds to a complex between polymer and nucleotide complex. And this avidite have a huge affinity, much stronger affinity for the complex with the polymerase and the chain of DNA. And so you decrease the volume of reagents that you need, that are needed for the sequencing, and a large part of the cost for the sequencing, when you consider the Illumina sequencing, is coming from the cost of the reagents, and the fluorescence is particularly expensive. So having with the avidit the capacity to bind at nanomolar rather than micromolar makes a huge difference, and at the same time you decrease the quantity of reagents, so you have a clearer signal to noise ratio, and so that leads to better, lower indel errors, higher accuracy, and cheaper price. So this is also interesting to work with some difficult regions. And so there is a trend at the moment for a lot of people to move from the Illumina sequencing, which was in a sort of domination that was difficult to leave for the different platforms. And so people have looked at the alternative. The first alternative that was looked was the BGI model, and you have seen that they are showing a very cheap price. Unfortunately, it was associated with difficulty to get reagents. So I don't know if other people here have a different experience, but in France it was really, really difficult to get access to the reagents. And so the AVT is developing a new alternative. And when you look at the annotation of the index is low. The comparison for the misassignation of homopolymers is very low when you compare with NextSeq. So the systematic errors are much lower. If you compare here, this is AVT, and here this is again with NextSeq. And the mean base quality, when you look over the sequence, so you can generate sequence over 150, and you see, but in two directions, and you see that we can get a mean base quality that is over 30 by comparison with 40 that you can generate with the NextSeq. So it seems really that there is some interest in this technology, and you can get it at a cheaper price than with the NextSeq. So the other part of the story is about the single molecule sequencing, and with that is introduced a third generation of sequencing. So there was a first trial by Stephen quake, and it was not successful with a company that was called Helicos. So I can go very fast with that. And they stopped in 2012. And the next player has been PacBio. And PacBio introduced a new platform that was called a single molecule real-time platform. And so this system they use microfabricated nanostructure. So you have a small hole in which you that they call zero-mode waveguides, and so they are made in a metallic film that cover a chip. And what happens is that when light is passing through very narrow diameter aperture that correspond to a diameter that is smaller than the wavelength, in that case there is a decay, an exponential decay of the light. And when you are using that, you can get a signal only from the very bottom of the wells. And so that allows the visualization of just the fluorophore molecules that are close to the bottom of the well. And so if you have a DNA polymerase inside the MW, you can get the extension of DNA chains and the incorporation of fluorescent nucleotide that is monitored directly and only specific to this bottom of the well. And so that gives you information not only about the binding, but also the time course of the binding. So you have kinetic data, which can be interesting to monitor information about the modification of some bases. And the technology is compatible to read a sequence up to 10 kilobase in length. And so that's particularly useful for de novo genome assemblies. And what was introduced by PacBio was to improve the quality of the sequencing. What they did is that they add some sequence in order to have a circular sequence. And so they were able to perform the reading of the sequence several times. It is always the same notion that is used. And so instead of reading only one time, you will get it several times. And so you improve the quality of the reading, and you can get very high accuracy. So when people are saying, well, PacBio is much better than nanopore, that we will see afterwards, this is just because you are reading several times. If you are taking several times read that is read with nanopore, you can do the same type of correction. But at least here, you know that you are working with exactly the same molecule, which can be important, and that makes a slight difference with nanopore. So the sequencer, the genomic sequencer, are fond of PacBio, and I've used it for a while, even though the technology was expensive. But it was used in many different studies, and especially to resolve complex structure in genomes. And so the last actors that we could introduce come with the technology of ion pore sequencing, and the first representative of it is Oxford Nanopore. So this technology was in fact invented when Kasianovich and colleagues found that it was possible to measure the transport of RNA or DNA through a bilayer through a molecule, an ion channel from bacteria called hemolysin. And then they, Dimmer and Akeson, just before 2000, validated that the current signals were produced by a single nucleotide and that they could be used to distinguish different nucleotides. And so then there was several developments, but the most significant were around 2010 when Oxford Nanopore Technology developed the mean ion, then the gradient, then the prometheon to allow the analysis of the signal that was crossing the nanopore. And so what was quite successful was the capacity that we have seen yesterday, that you can come with your your sequencer and you can run. Can you say how many reads we have today?
2.3 million.
So congratulations.
I think it's your order because this is more than double the best run I've had before. So I'm attributing it to you to the fact that you sat next to it. And all of those years of experience and wisdom made the device run a little bit more efficiently.
We will see if the sequence is correct. and we are waiting for the winner, the one that will decode. Okay, so we have already seen what was possible, and besides that, there were funny use of it in the field. For instance, Lohmann sequenced Ebola viruses directly in Guinea. It was going into the sky, into the deep ocean. It was a lot of media were involved. So the fact, well, I have perhaps no need to develop the function of the nanopore because we discussed it a little bit yesterday. So we have a protein channel that is embedded into a lipid membrane that form an electrical connection between two salt solutions. And so if you have an electrostatic potential that apply between the two sides, you will drive some single-stranded DNA molecules through the nanopore aperture. And so there is additionally a DNA helicase that is bound to the DNA to control the single-strand DNA movements for the DNA. And so that feeds the nanopore one nucleotide at a time. And so there is a lot of work to be done for analyzing the signal, and probably we will discuss that later on. But what is important is that there is a major operator in this area that corresponds to Oxford Nanopore. But concurrent to that, there are several companies that are developing their own system. So BGI is developing a system called CycloneSec, which is nearly exactly the same, except that they have modified the pore. They have modified the configuration in order to allow a higher amount of pore to be inserted into the membrane. So this is a novel chip design, and they are also developing some novel Bascoling algorithm. But what is interesting in the case of the technology, and that can be true for Oxford Nanopore as well as for the CycloneSec, is the capacity to make long duration sequencing, having a high generation of data, and what is particularly attracting is the capacity to have very large average DNA fragments. You see N50, 50% of the read can be above 33.6, and a nice mapping in the case of human genomic DNA sequencing. So there are a lot of things that needs to be done in terms of the signal analysis. There are different types of base calling, and maybe there are better specialists than I can be to discuss that, but what Nanopore is proposing is a different type of base caller. So the latest one is Dorado, that we are not necessarily using all the time because at least in our hands it has been super slow, but it can increase quite significantly the quality of the score, the accuracy to 99.75%. And so we are getting some quite good data with this type of technology as well now. So this is just an illustration of the cyclone-seq. And what is also quite interesting in the case of the nanopore, but which is not necessarily directly in line with what we do in MoleculArXiv, is the capacity to detect also native modification, and that is becoming more and more accurate in the case of this Oxford nanopore. They are in particular working constantly to expand the repertoire of detectable modification of DNA and RNA. And so you have here different modifications, so methyl 5C, methyl 6A, you have inosine. So I think that this is, in that case, this is RNA, but you can have also a direct identification of the methylation of DNA and hydroxymethyl. So that makes a particularly interesting aspect of the work with the Oxford nanopore, and people are actively involved into that. I wanted just to finish with beside nanopore. So Roche is coming back, and they have acquired a company that is called Stratos. So I don't know for the quality of the output, but the quality of the I don't know if I can. Yes. The quality of the imaging is quite amazing when Roche is doing something. So the idea behind Stratos is to have the incorporation of a different type of molecules. So instead of having the inclusion of a nucleotide in the chain that you want to measure, they are introducing what they call expandimer that correspond to a very long chain that correspond to several times the same nucleotides. And so the advantage is that you will have a very stable signal to detect a C, to detect a T, to detect a T, to detect a T, and okay, it's too fast. So they are claiming that the technology will be very good. So there is a paper in bioRxiv that has been published, and there is also a description in the patent that you can access. And so the machine is a big machine. You have here an illustration of that. It will be an expensive one, but they are claiming the possibility to scale to the size of 8 million nanopores, and probably with an output of 500 million base per second. Well, you have to consider that at that time, they are only at 280 base per second, so they have some improvement to make, but they are claiming that it's easy to do. And so in the near future, it may be that this will find a space for some type of sequencing but it is too early at the moment. The machine exists, however. It has been presented in a congress, the Congress of the Human Society of American Society of Genetics, Human Genetics, and so I think that it will be probably available in the next years with I think a cost of above 1 million of euros per instrument. So I will not spend too much time on the technology like the one developed by MGI, the DNBSeq, or there is also Ultima Genomics that is developing another platform. What is interesting in that case is that the very, very high throughput is possible. And this is the type of instrument that is raising the individual genomes below the cost of 100 euro per genome. So making it a particularly valuable instrument, but only if you have a huge throughput. So it's probably in clinical context, or unless we develop some application in the field of. But what I think important is that if you develop the next generation of synthesis, it will be also very important to develop the capacity to read not only some gigabytes like the human genome at 3 gigabytes, but also some capacity at tera or petabyte, and I don't know what it is at the moment that can be doing that. So there is a very important issue about that, and I think that it is one of the challenges that will be facing, that will be necessary to face for MoleculArXiv and all the people working in the field. Okay, so yeah, for the analysis, well, I have very basic description, just to mention that there are more devoted to the analysis of reference genome. What we are interested in the case of DNA storage, is to be agnostic and to detect the sequence that we don't know. So the base calling, the demultiplexing, all these sort of things can be difficult to consider directly. The generation of FASTQ file, the demultiplexing of pool sample, all these sort of things can be done, and with some primary analysis we can generate information about that. We have some specific tool depending on the different technologies. But there is something very standard. We can generate some read quality assay by FastQC, MultiQC, these sort of things. And so there are specific requirements. For instance, if you are working with circular consensus reads, in the case of PacBio, but the companies are usually developing their own software, and usually they have been optimized over the years. So the quality score, we already mentioned that with the idea of measuring the Q score, and so I stopped to the 30 because it was the standard for Illumina, but I should come to the 40 and even the 50 in the latest sequencer. So it means that the error is 1 out of 10,000 or 1 out of 100,000, and that makes a big difference when you want to do any kind of analysis. And so the alignment to a reference genome is not well. We are doing it when we are doing some analysis for data storage, because we know the sequence of the oligos, but this is not the way it should work, and this is only during this first phase. But we have also to develop our tool for that. And just to finish, for the format, I can skip it. And yeah, I wanted just to, the kind of output that we can generate is illustrated by this plot coming from IGV, in which we have seen it is just a representative oligo from several batches of synthesis that have been made. So that's a long story and a sort of difficult experiment that we had with a synthesis that was done by GenScript and that was not successful. So the kind of data that it introduced was some errors in the sequences that were not necessarily detected with nanopore or Illumina. And this is a second run of synthesis that was generated by GenScript, in which it worked well. And well, it's not too bad. It can be worse here, because you will see that there is a lot of deletion in the middle of the sequence, so we cannot interpret correctly the system, so the signal, while we can save it even though we have some deletion in the middle of the sequence. And here we have some very nasty sequence that was generated during a second run of synthesis, so when it was much better. But the first run was totally unsuccessful, and we were even unable to get a signal with Illumina sequencing. So that's the type of things that we can monitor very easily, and then all the different assembly are under the hand of our collaborators. But you see that having this sort of signal that are directly explained by an error during the synthesis of the oligonucleotide that is missing totally in the second run of synthesis can create big issues. You can see that with nanopore and with Illumina. And well, sometimes the most difficult part is to convince the provider of oligo that the mistake is on their side. It was for us difficult, because when we contacted them, we had only done some nanopore sequencing. They said, well, sorry, we are only checking on Illumina, so too bad for you. And then we came back with this sort of pictures, and they changed their mind. That's it. So, well, at the end of this presentation, the quality of the synthesis is really crucial. So it's not in our hands, but definitely what is needed is also the development of easy-to-work sequencing capacities. So that's interesting to have the nanopore, because it is, as we said, easy-to-run solution. But as I said also, it's very important to go over that and to develop the capacity to read much larger sets of data. And so we need to consider all the different options in order to have this sufficient level of sequencing. And with that, I'm finished. Thank you.